Yeah. Again, I'd like to thank the Food Packaging Forum for inviting me to speak. And I'll just give a little background about myself. I am in the National Exposure Research Lab, which is part of the EPA Office of Research and Development. So we are the research arm of the agency, and we do the research that provides the data and models to the regulatory part of the agency to aid them in their decision making. So I'm going to be talking a little bit, I'm going to give a little bit of background of the challenge that we're facing in exposure assessment of chemicals, and talk about some of the modeling we're doing on food contact materials and kind of how we ended up even working in that realm, and then talk about the impact of that model in our overall framework for decision making on chemicals. Okay, and I'm required to state that the views expressed are mine and my co-authors and don't reflect the policies of the agency. Okay, so in the United States, different chemicals that have different uses are covered by a tapestry of legislation and regulatory and testing requirements. Pharmaceuticals and cosmetics and food additives are handled mainly by the Food and Drug Administration, while pesticides are handled by a particular group within EPA. But most other chemicals, industrial chemicals and dye and many consumer product chemicals are covered under the Toxic Substances Control Act, which is administered by EPA. And this TSCA was passed in 1976, and thousands of chemicals on the market were what we call grandfathered in at that point with no testing. Thousands of new chemicals also come through our new chemicals program on the regulatory side every year. And so TSCA was amended a couple years ago to allow what we call risk-based evaluation of these existing chemicals and new chemicals. And so methods are being developed within the ORD under our Chemical Safety for Sustainability Research Program to inform prioritization of these existing and new chemicals for testing. So I wanted to mention that this is kind of a figure from the New Scientist magazine a couple years ago, and this ends up in almost all of our presentations. And this is really, kind of distills down the problem from the agencies. We have thousands of chemicals that EPA administrates the regulation of, but we don't know which ones are the most important to look at first. Okay. So when we say high throughput risk-based prioritization, you can kind of see this is the workflow that the amended TSCA requires. And so you can kind of argue about the actual number here, but there's approximately 70,000 chemicals in the TSCA inventory that have to go through this prioritization process. And the chemicals that are the highest priority will undergo an in-depth risk evaluation. And so I co-lead what's called the ExpoCast project. So I co-lead the group that is looking at the exposure component of risk, which needs to be combined with the hazard side of risk in order to make any kind of risk-based decisions. So the idea here is that from bioassays, from either the ToxCast or Tox21 program, we have some idea about the intake dose of milligrams per kilogram body weight per day that would be required to elicit some type of biological effect. And then we can compare those with the exposures that are coming out of our ExpoCast project. And this process is most meaningful when we can quantify the uncertainty in both hazard and exposure as well. So all these methods are uncertain, but if that uncertainty can be qualified, we can make informed decisions. So no model is perfect and may have high uncertainty, but can still be used to be fit for purpose for some type of decision making, such as prioritization. So you can see here chemicals, even though they have high uncertainty, both in hazard and exposure, if they're still several orders of magnitude away, that's a less interesting chemical. And chemicals may overlap here because they have much higher uncertainty. And then this is a very interesting chemical where your hazard and your exposure have fairly small uncertainties and overlap quite a bit. And so this is kind of what we're doing in our project. Okay, and so why is EPA even getting into food contact, right? So the TSCA legislation says that this prioritization process must consider conditions of use, which are the circumstances under which a chemical substance is intended, known, or reasonably foreseen to be manufactured, processed, distributed in commerce, used, or disposed. So we have to look at many different types of exposure pathways in order to look at all conditions of use. And so In the exposure science realm, we have the idea of an exposure pathway. And so that's the course an agent takes from the source, the chemical source, to the target, which in this case, we're talking about humans. So we have manufacture, we have creation of consumer products, we have fate and transport in the environment. And this is really what our ExpoCast project is getting into, is trying to understand exposures via all these routes. And so we are building models for consumer products, for what we call far field exposures, which are industrial releases from pesticides, and ultimately food packaging, I'm going to be talking about today. And so how do we integrate? We integrate information from all these different models for pathways by evaluating and calibrating them with biomonitoring data. And so we do that in a framework that we call the systematic empirical evaluation of models, and it's described in 2 papers by my co-lead on the ExpoCast project, John Wamba. And I'm not going to really get into the weeds here, but basically what we do is we take predictions from all these models and we perform a linear regression. We use these models as predictors to see if they can predict biomonitoring, right? And it's kind of like a sensitivity analysis. From this regression, we can determine which models are working and where more data would be most important. And this is just an analogy that those of us in the southeastern United States are at least are familiar with, that predicting, another consensus type modeling approach is hurricane path prediction, right? So you have models, all different kinds of models. We have US models and European models for predicting hurricane pathway, and then you judge these models based on how they perform historically. And we're doing basically the same kind of thing here with our models. Okay. So just to be clear in terms of our goals and the goals of other agencies, what are we doing at EPA? Well, we are trying to understand total exposure potential across many different pathways for tens of thousands of chemicals using high-throughput models. What we're not doing is any in-depth exposure risk or safety assessment for individual chemicals. So that's really our domain, is prioritization. Okay? So now I'm going to talk about the actual food modeling that we have, food contact modeling that we have done. And so very, very simply, it's really quite simple. It's at heart, it's a bookkeeping operation. So you have concentration in the food times food consumption or food intake gives you an exposure. And so how we tackle each of these, or how we tackled each of these, was for food concentration, we actually built an empirical model of migration from food contact materials based on some data, a data-driven model, and we can use that model then to extrapolate and predict concentrations for food chemicals, a variety of chemicals in food contact materials for 12 different food groups that have different properties and thus have different migration. For the food intake, we actually take consumption diaries from the National Health and Nutrition Examination Survey and Haynes, What We Eat in America. And so this is a nationally representative study of food consumption in the United States. And then we map they have 7,000 different food codes in this study, and we map that actually onto the same 12 food groups for which we're calculating migration. And then we have what we call an exposure model, which is basically a large population-based simulator where we use US Census data to predict predict the US population by age and gender and food consumption. We match those diaries up. And this work is described in a paper in Environment International, which is first authored by Daria Burial, who is my postdoc. Okay, again, not to get into the weeds too much, we've seen already this morning kind of this system, the polymer food system, where you have these really key parameters of the initial concentration, a diffusion coefficient, and a partition coefficient. But we're looking at tens of thousands of chemicals here. So mechanistic and deterministic migration models are really not that apply to tens of thousands of chemicals and lots of different foods and different polymer systems are not really available. So we used an empirical approach. So we used some data that were obtained from the Food and Drug Administration. These were migration experiments with different food simulants, different temperatures, different chemicals. And 436 of these experiments, we could say, had reached equilibrium. So we built an empirical model linear model for log migration as a function of all these things which we know impact and exposure. Okay, and so this is what the results of this model look like. Based on our predictors, we were actually able to describe about 70% of the variance among chemicals from this data set. And on the right-hand side here, you can see which chemical properties and food properties actually were driving migration. And so these are things that are are reasonable based on what we know about the mechanistic properties of migration. So for higher initial concentrations, higher log of the octanol water partition coefficient, higher temperatures, higher water solubilities, higher solubilities of chemical and water, you see higher migration. And then compared to fatty foods, foods that are alcoholic, acidic, aqueous, have reduced exposure. And then we also have reduced exposure for molecular weight. And so again, this is a simplified model, but it's a model that, even though it has large uncertainty, again, we think that this could tell us something in our high throughput approach. And because we can characterize uncertainty, this model could still be used to make fit for purpose decisions. So again, not going into it in too much detail, but that migration model then can be extrapolated to other chemicals. So we actually use the migration model to predict concentrations in over 1,000 chemicals that we saw we identified as being in food contact materials from FDA and European Union lists. We had some C0 information value or initial concentration information from the literature, but we also estimated some based on the data in the actual database that we got from FDA. And we were able to convert, and we calculated concentrations in these 12 different food groups. So different combinations of temperature, food storage temperatures, and food type. And then all that's left to do, again, is to calculate a consumption. So again, not go into this too deeply, we took the food diaries, mapped every single food code to one of these 12 food groups based on food temperature and storage, and that gives us a diary for each individual. And then that information was incorporated into our exposure model and sampled. So we simulated a population of 35,000 people, representative of the US population, and each of those people got a food diary, and each of those foods in those food diaries had a specific concentration associated with the chemical, and we can get a distribution of exposures. Okay? And so I was asked to talk a little bit just about our key assumptions and uncertainties. So here we have kind of some standard assumptions that generally apply when we're doing migration modeling. We're assuming a monolayer and the packaging is in contact with the food. We're only looking at storage, we're not looking at heating, and we assume that migration into dry foods was assumed negligible. But really what I wanted to point out is that we really had a lack of information on the initial concentration of the chemical, and so that was a really key parameter that drove migration. And the current method of estimation that we're using, which was basically using the functional role that the chemical is taking in the packaging and making some assumptions about what weight fraction that might mean, imparts critical uncertainty in our results. And there's also the model uncertainty. This is a very simple linear model. Models that allow us to predict new methods to actually build QSAR models for diffusion coefficients and partition coefficients would better reflect true migration patterns. And then we also need better ways of understanding consumption factors. We had to make the assumption that all of our model chemicals occurred in all polymer packaging, and that we had no chemical-specific consideration of market, polymer type, or food type distribution. So which types of chemicals are in which types of polymers that package which types of food. Okay, and then finally, I'm just going to finish up very briefly with the impact that this model is having in our overall modeling framework for exposures to different types of chemicals. So we have a large multi-organization collaboration to bring lots of different pathway-specific models into our SEAM evaluation framework, and one of these being this high-throughput food contact model. And so what we see that when we brought all this into our consensus framework, and this is a Bayesian multivariate regression against biomonitoring data, it turned out that chemicals, the chemicals that had food contact pathways and consumer pathways had the highest exposures relative to the mean exposure in NHANES. And this food contact model was the only predictor. We were treating models as predictors. It was the only predictor that was significantly predictive of exposures for the dietary pathway. And so what we can do then, this is our consensus model, and then we can apply it to all the chemicals for which we have data, for which we have model predictions, and for which we have chemical properties. And what this turns out, that we've actually now been able to apply this model to about 687,000 chemicals. And we can basically, what our framework gives us is the uncertainty as well. So even though these are the median exposures, these are the upper and lower 95th percent bounds on that median, we only have about 1,976 chemicals that had exposures, 95th percentile exposure greater than 0.1 milligram per kilogram per day. So these are our most interesting chemicals. So just to conclude, we have to prioritize thousands of chemicals. We're building high-throughput models to do that. Even models, high-throughput models with high uncertainty can be meaningful in certain contexts, and they can still be fit for combination with high-throughput data for prioritization. That's it.
Thank you very much. You covered a lot of material in 15 minutes.